I go to school in America called Portsmouth Abbey School, which is located in Rhode Island. And I chose my English name Scarlett because I was really impressed with the movie Gone with the Wind and the character Scarlett O'Hara. And in my spare time, I usually spend some time with my friends doing some performing art, including theater and dance.
Okay. Hi, I'm Matthew, and I'm a rising junior from Seoul, South Korea. And I go to school in Seoul, South Korea. It's an international school. And something interesting about me, I've never drank coffee before. And also, I was trying to think of some hobbies, and the first two that came to my mind was tennis and food competitions. So like, where you go to a restaurant, they have like a challenge where if you can finish something in 30 minutes, they'll put your name on the wall, or things like that. I like going around Korea and doing those.
Wow. Do you know Nathan hot dog competition?
don't do that in America.
Yeah, I love eating competitions in America.
Yeah, Nathan hot dog. They're idiots.
Okay, so I think we should move on to our research that we did before we. So as Professor Kim mentioned before, Mitchell and Jonathan were working on TLF for a while. So we tried to jump into that project and see how that specific technology as fluorescence is a viable option to measure the water quality accurately, and also if there is any correlation between the new technology fluorescence and the old and traditional but well-used methodology, which is the log plate counting. So the first question that we have to face was, why do we care about the water quality? So because the microactivity that is indicated, that is shown in the water, is the main feature of the water quality. And that microbial activity finally leads to the open defecation and contaminated water, which is the main cause of the disease that's happening universally. So we narrowed down into two main objectives of our research. So first of all, we wanted to try to figure out what is there any relationship between fluorescence and plate counting, and also we tried to figure out if the cheap and portable thermometer that Mitchell and Jonathan are working on is a viable option. So there are three main methodologies that we have used in the experiment. The first one is the Delagua, which I mentioned before, the traditional one. Second one is spectrophotometer, and the third is the main innovative one, spectrofluorophotometer. So I wanted to give you guys like a brief overview for the Delagua process. So first of all, you have to set up all the plates, which are sterilized already, with the sheet that can soak all the lauryl sulfate broth that has the nutrients that bacteria can grow. And you put the water sample into the cap of the tube, which is you can determine if you want to dilute or undilute, but you put the water sample and put the filter membrane in between, so that and by pumping squeezing the pump, you can extract the water from the water sample, so that only the contamination can remain on the filter membrane. And when you take out the filter membrane, you put on top of the sheet, which already soaked up all the lauryl sulfate drops, and you put the stack of plates into Delagua heat for 18 hours to incubate. And when you open it after 18 hours, you can see the number of colonies that are already formed. So these are the pictures that we have done in Delagua for 4 weeks. And we thought these are two main parts of Delagua. So we thought the first part is really important, which is filtration with the filter membrane by creating the vacuum with the pump, and also counting the culture coliforms, getting nutrients from lauryl sulfate broth. So even though Delagua is really old, traditional, and well-used methodology, we thought there are like really big problems, weaknesses for the methodology, so that we can move on to the spectrofluorophotometer. So first, like the pros for Delagua is it's giving us quantitative data, which is the exact number of colonies that are formed on the filter membrane. So that can lead to the high accuracy of the data points, and also because it only requires a Delacqua key, it can be done in the field. But the four main problems of the Delacqua is it's really time intensive because you need at least 18 hours to incubate the plates with water samples, and also because there are a lot of parts for Delacqua key, it's a large amount of equipment to carry. And because it is done by humans with all the equipment, there's a lot of potential for contamination, for example, not sterilized equipment. And also, you choose whether you're gonna dilute or undilute the water sample, there is a chance of using a wrong dilution factor.
Okay, moving on to our second technology. So the second technology we looked at was the spectrophotometer. So the main reason we did use the spectrophotometer was to measure the absorbance so we could calibrate and correct our spectrophorophotometer measurements, but we also did consider it as a viable alternative to the spectrophorophotometer in measuring water quality. So this is a diagram of how a spectrophotometer works, and what it does, to put it simply, is it just measures the absorbance of light at specific wavelengths of a sample. And so a light source shoots light at a mirror which is at a specific angle to let only a narrow band of wavelength of light through the slit, which goes through the sample, and then the detector reads whatever goes through and therefore can measure how much of it didn't get through and was therefore absorbed. And before we go to our third and main technology that we looked at, the spectrophorophotometer and TLF, I just want to do a brief overview of the science behind the technology. So fluorescence, as many of you probably already know, fluorescence is a process of light excitation and emission. And so a molecule and electron in ground state is hit by a certain number of photons that allow it to go up to an excited state. After a while, it drops back down to the ground state, and in the process, releases energy in the form of photons. And the important thing to note here is that the emitted light is at a lower energy and therefore of higher wavelength than the excitation light.
Okay, so scientists figure out that tryptophan is a good indicator of microbial activity that happens in the water. Tryptophan is basically a kind of amino acids that has indole group, which is distinguished with different types of amino acids. Indole group is the part that leads to the fluorescence property of tryptophan, because when tryptophan absorbs some energy, the light from other photon or other light source, it emits the fluorescence, so that indicates the microbial activity.
Okay, so TLF. TLF stands for tryptophan-like fluorescence. And the reason we're focused on this is because we employ the unique fluorescence properties of tryptophan. So what I mean by that is tryptophan, when hit by 285 nanometer light, emits light at around 340 to 370 nanometer light. And well, we specifically focus on 360 nanometers, but this allows us to distinguish it from whatever else is in the water. And tryptophan since tryptophan is organic, it's been linked to organic matter and therefore, to a certain degree, microbial activity in the water. And so it's important in measuring water quality. So this is an excitation emission matrix that we made of 250 parts per billion tryptophan solution, just to give a visual representation of what exactly is in the solution. So this band is caused by Rayleigh scattering, which wasn't that important in our research, but what Rayleigh scattering is when molecules are hit by light, it scatters the light without any loss of energy. And the important part here is this band of colors, which is caused by TLF. We know this because of the excitation wavelength, which is 285 nanometers, and then the emission wavelength, which is ranging from 340 to 370, focused around 350, 360 nanometers. So moving on to the actual technology, spectrophorophotometer. So what it does is, as I mentioned, it shoots light at a sample and it measures the resulting intensity of the emission light at specific wavelengths. Yeah. And so some of the pros and cons of the spectrophorophotometer versus the traditional methodology, the Delacqua with plate gallium. So the biggest pro we found was that this is a lot faster than Delacqua. especially compared to Dalaga and plate counting's 18-hour incubation period. It's basically instantaneous. Just click a button and your readings pop up. And also, like Dalaga, it provides quantitative data. And some of the cons of the spectrophotometer we found was that, first of all, the readings of the intensity are instrument specific. What I mean by that is when the computer reports and the machine reports the intensity of the light, it does it in arbitrary units that vary slightly across different machines. And also there are various inherent factors interfering with the intensity readings, such as quenching, inner filtering effect, which we will mention in later slides. And the third con we found was that, well, for us at least, our equipment was quite cumbersome and expensive, and it really couldn't be used in the field like Dalaga can, which Mitchell and Jonathan were trying to fix in their research. So, like I mentioned with my first con, readings are instrument specific. And to fix that, Raman scattering. So what exactly is Raman scattering? Raman scattering is the scattering of light, mainly by water in our case. And the thing that differentiates this from Rayleigh scattering is that while this is also rare, some energy is lost, resulting in a peak at a slightly higher wavelength. And we use this to calibrate our readings for our fluorescence intensity, because across different labs, we use DI to make this graph, and since all DI is the same, this area under the curve of the Raman peak is also the same across different machines. And so we can use that area and we divide our readings by that area to calibrate our readings. And also, the second con I mentioned, quenching. Quenching is just anything that interferes with fluorescence intensity. And quenching happens through multiple factors such as temperature, pH, colloid concentration, collisional quenching, metal ions, and oxidation, but mainly temperature and colloid concentration. And we focused on the effect of temperature quenching. And so we made 3 different L-tryptophan solutions at 50 parts per billion, 100 parts per billion, and 200 parts per billion. And we looked at the effect of temperature on the fluorescence intensity readings. And we found that, as you can see, across the board, no matter the concentration, as temperature went up, intensity would go down in an inverse relationship. And we also use these findings and this graph to later correct our different readings for temperature.
So another source, excuse me, another source for error of intensity reading is inner filter effect. So when the inner filter effect happens, when the light source emits the light to shoot the sample, some light that emitted is reabsorbed by the other substances that exist in the water, and also the TLF, the fluorescence that is emitted from tryptophan, is also reabsorbed by the quenchers, so that disturb the intensity reading. So according to the literature that we have read, it says we have to multiply the correction factor to the intensity that is collected with the Raman units. So this is the equation of I factor. And then we also plotted the graph to compare the correction and uncorrected data points with 3 different samples. And as you can see, before we corrected the intensity reading, it was slightly low because the light was reabsorbed by the quenchers. But after we corrected, it gives us accurate data points. So we tried to connect the intensity into the concentration of the L-tryptophan. So when we run the experiment with L-tryptophan solutions, we don't really have to care about the concentration because we already knew about the concentration because we made it. But if you put that into the real field of collect a water sample from the nature, you really need to think about how you can convert that water sample's fluorescence intensity into the concentration of L-tryptophan. So we plotted a graph to see the correlation between intensity and concentration. So the X value is concentration in parts per billion, and the Y value is intensity at 360 nanometers in Raman units. And as you can see, it gives a strong linear relationship, which has R-square value as 0.987. So actually, we're curious about what's going to happen with a higher concentration of L-tryptophan. So we made 250, 500, 750, and 1,000 pp parts per million concentration of L-tryptophan solutions. And as you can see, with the lower concentration, it gives a linear regression line. But as the concentration goes up, the graph levels up. That means it doesn't really change the intensity. So before we move on to our data points that we have collected over 4 weeks, we want to give you guys a brief line for the water sample size that we collected. So of those, the Mill River and West River were the main sources that we got the water samples, but mainly focused on the Mill River. So we collected from 7 different parts of Mill River, and these are the names of the water sample sites, and this is the picture that Mitchell took from West River. So before we combine those 2 data points from fluorescence reading and plate counting from Delaware, we wanted to see how trends gonna look like for each respective data points. So we plotted the graph for spectrophotometer intensity reading and Delaware plate counting. And as you can see, the trends are really similar, giving increasing slope, and also because it's the water samples are in the order of upstream to downstream, it has the correlation. We can see somehow it's like connected with the location of the water samples and the data points. Also, because we ran the experiment with spectrophotometer, we wanted to see how that trend also looks like. So we collected the data points at UV254, which is the point when organic matter absorbs the light at most, and this is the graph of absorbance at different water samples.
Okay, moving on to the main part of our results. When we started out our 4 weeks here, one of our main goals, our main goal was to see if there's a relationship between Delaware plate counting and TLF on the spectrophotometer. I think we did find a relationship. So at first, we were both kind of disappointed by the R-squared value because we thought it was a bit low. But then professor told us that given the circumstances and like the experiment itself, the R-squared value was actually high enough where we could conclude a statistically significant relationship between the two.
Oh, I said it was not that bad.
Not that bad. Okay, close enough. Okay, and we thought that maybe the readings on the Delaware plate counting were slightly off because, first of all, some of our dilution factors, we messed up on some of those. And also, when we were counting the plates, sometimes we would only count a quarter and multiply by 4. And so multiplying by 4, multiplying by the dilution factor, all of the multiplication probably threw off the readings quite a bit. Yeah. And also, this is just the graph where we converted the y-axis TLF from Raman units to concentration in parts per billion, just for ease of viewing. Yeah, so more out of curiosity than anything, we also tried plotting the relationship between Delaware plate counting and UV254 from the spectrophotometer. And like Scarlett said, the reason we use UV254 is because that is the wavelength at which benzene organic matter absorbs the light the most. And we were surprised by the R-squared value and the relationship because it was actually stronger than our main focus, the spectrophotometer. And we weren't sure if this was just a coincidence because of the lack of data points, or if there's actually a better relationship between UV254 and plate counting than we thought. But in the end, we did decide that this did warrant further research.
So we made 3 main conclusions for our 4-week research. So as Matt already described about the correlation between fluorescence intensity and plate counting from Delaware, we concluded that its spectrophotometer can be a viable option to measure the water quality accurately. And also, even though the spectrophotometer intensity reading is quite fast measurement, it has so many confounding factors that can actually interrupt the reading, so that we have to fix.
Yeah, the third conclusion, like I already said, is that UV254 has an unexpectedly strong relationship with plate counting.
So we thought about how like future research topics gonna look like. So we're thinking of how about making some methodology that can correct the intensity reading, which has some errors on the spot, so that we can gather the data points with some accuracy and precisely. And also, we try to figure out if the portable fluorometer that Mitchell and Jonathan are working on can correct the data point on the spot. And lastly, we also, as Matt mentioned, we really wanted to explore the correlation between the UV254 and plate counting for Delagua, because it was so interesting for us to get the data, like R-squared value 0.8. So we wanted to figure out with more data points.
Yeah, and so some of the lessons we learned over our 4 weeks here. So first of all, we became able to construct and perform experiments by ourselves and work towards specific goals that we had in mind. And we also learned that Delagua and plate counting are really tedious processes and they take a lot of time. And we also learned never to drink or swim in the Mill River.
So we feel really thankful for Dr. Professor Jae-Hong Kim to give us an opportunity to experience the real science field, and also we really appreciate Mitchell and Jonathan's 40 hours a week and their effort to collect the water samples every week.
And we also want to thank the Environmental Engineering Department of Yale and the entire Kim Lab for helping us out, even in small ways. And we also want to thank Loris for making this whole thing possible and sending us to this wonderful program. And also, last but not least, we want to thank the Environmental Health and Safety Department of Yale for sending us our net IDs and PIN numbers in an efficient and timely manner, 2.5 or 3 weeks. Yeah.
Sounds accurate.
Any questions?
Thank you.
Great. Any questions?
When you guys presented the graph where you have the correlation between TLF and the Delagua, where the TLF values, you talked about correction factors earlier, did you correct those values for temperature and internal absorption?
Yeah, so we converted that to Raman units, we corrected for temperature, and we multiplied by I, and
So
we filter correct to that.
because especially the other factors weren't as significant, so we decided we didn't need to correct them, like pH.
Was there a big, did you see the correlation improve a lot between the correction and the non-corrected value? Or was it pretty similar?
We never actually looked at the relationship between the uncorrected and just regular plate counting. We corrected and then we plotted the graph. But we assume we probably would have.
Does it make sense that you see a correlation between the plate counting and UV254 absorption? Does it make any sense? What would be the reason?
But, yeah.
Because organic matter,
Yeah.
the whole concept behind TLF is that organic matter,
Right.
we correlated organic matter with microbial activity.
Right.
And UV254 is, as we said, the wavelength of light at which benzene organic matter absorbs light the
Right.
most. And yeah, so I think that
That makes
positive
sense.
kind of makes sense.
Right. So
That's
dirtier water has more organic
Dirtier
water
has
germs,
organic
right?
matter, therefore.
Right. Why
probably
don't
more
you
microbial.
tell Jonathan and Mitchell throw away the idea of a spectrophotometer and just measure UV254? Have you discussed that with Jonathan and Mitchell?
Kind of.
What's your response, Mitchell?
We're not letter kings, guys. That sounds like.
Any other questions? Minjo, you should ask questions.
I don't know how it feels to be asked questions, so I'm not gonna ask them.
What would you say is like the major benefit, or can you think of what would be a major benefit of TLF over UV254?
I can't really think of anything. They're both pretty fast. UV254 spectrophotometer is slightly slower, but at the same time the readings are a lot easier to interpret, and they don't, as far as I know, require as much correction.
But in theory, the tryptophan, you are assuming that they originate from what? From germs, while other organic matter can come from anything, right?
I guess the spectrophotometer and TLF would be more specific.
It can filter out other organic matters.
Explain those photos.
So this is a picture from 2000 when I was really young. I was traveling with my family, and my dad always carried this picture with his wallet.
And this is a picture I cropped out of Facebook.
Which one is you?
This one, right? This guy right in the middle. So this was back in 5th grade, and I was going through my emo phase, and I thought I was super cool. Yeah.
No more questions? Come on.
So I haven't, because I actually haven't asked you, what was your favorite experiment to run out of all the different methodologies you've been using, and why?
Actually, even though it's really tedious, I really loved it. Yeah, it was so fun to like count all the colors like with yellow and like pink colors. It was
That's how much
pretty
you said
fun.
when we're doing the experiments.
Stephanie, are you still enjoying
What? Yeah.
counting?
Oh, am I still enjoying plate counting? Yeah. Do it 3 years. It is satisfying though to like have grown and you can see it, and that's very tactile. It's really there.
Yeah, I also spent quite a lot of time plate counting, and for a few years after that, I never even touched Jello.
It's fun until you have to clean up.
Oh yeah.
Right, but thinking about like these high school students came here without any prior knowledge in this area, and within 4 weeks, come on, just 4 weeks, they were thrown at the beginning with just bunch of literature. They have to learn all these basic concepts, and perform this much experiment actually with a graph, actually real graph. That is amazing. So another round of applause for them. And also another round of applause for Mitchell and Jonathan. Thank you so much for all the hard work and great supervision to save graduate student time. Otherwise, someone else could have done that. Okay, great. Thank you. All right, thank you all for coming. All right, so that's it.